It's not, people say, oh, everybody knows glutamine. Yeah, but everybody misinterpreted how glutamine was driving the cancer. We have shown that it's a fermentation metabolism, not an oxidative metabolism. And that's a big difference for those that are in the field of energy metabolism and cancer. There's two ways to get energy in our body. One is an ancient substrate level phosphorylation. When I say phosphorylation, meaning you add a phosphate group to ADP to produce ATP, adenosine triphosphate, which is the chemical energy equivalent. The transfer of the phosphate to proteins and lipids and things like this changes the configuration. It is energy. Where do we get our energy from? How do we make ATP? We eat food. Okay. The energy of the sun creates carbon hydrogen bonds originally in the food that we eat. Ultimate, all energy comes from the sun. And then when we eat the food, we break those carbon hydrogen bonds and release again the energy of the sun and capture it in ATP. Without ATP, no cell can grow or survive. So the question is, how does the cell make ATP? And there's two predominant mechanisms of making ATP. One is an ancient pathway called substrate level phosphorylation, doesn't require oxygen. And this is the kind of energy that all cells had in the evolution of life on Earth. All life on Earth started from single cells that were using this substrate level phosphorylation because there was no oxygen in the atmosphere at that particular time. Only after cyanobacteria started to produce oxygen did we start to get more complicated structures, multicellular organisms, and eventually us and other mammals and fish and what we see. But every one of us, we're all getting energy from ATP. Well, over this time, these ancient pathways have become minimal. They play an extremely important role in our energy metabolism, as you and I are sitting here breathing. And we're getting energy from oxygen. But the preparation of the fuel source is using these substrate level phosphorylation as a very minor process. But when the when the ability of the cell to get energy from oxygen becomes corrupted in any number of ways, the cells gradually fall back on these ancient pathways. And this is what the cancer is using. It's called substrate level phosphorylation. Warburg described it in the cytoplasm. He received the Nobel Prize for actually defining the protein that actually gets the energy from oxygen in the mitochondrion. But he said these cancer cells aren't using oxygen. They're using this ancient fermentation pathway. And he tried to quantify how much ATP was coming from fermentation versus oxidative respiration. That was where he made one mistake. We now know it's not possible to accurately quantify where the energy is coming from. Our big thing is that we also get energy out of that same organelle, but using an ancient fermentation pathway embodied in that organelle, so the mitochondrion. So the mitochondrion in the cytoplasm produces most of the energy. And that's where we're breathing, because the mitochondrion takes in the oxygen and uses it as an acceptor so we can get energy through oxidative phosphorylation. Highly, highly efficient. Highly, highly efficient. Substrate level phosphorylation is inefficient. You have to use a lot of substrate, a lot of material to get a small amount of energy. But the cancer cell has fallen back on these ancient pathways because the organelle that generates the high efficiency energy is no longer efficient. It's inefficient. So the cell uses ancient pathways to grow and reproduce. And the organelle that controls the differentiated stable state of the cells in our tissues, that organelle is the mitochondrion. It's the controller of the differentiated quiescent state. And when that organelle becomes corrupted, inefficient, it falls back on the fermentation and loses the control of the growth of the cells, thereby leading to dysregulated cell growth. Dysregulated cell growth is the definition of cancer. Cancer is cell division out of control, dysregulated cell growth. How did it happen? From a chronic damage, chronic interruption of oxidative phosphorylation with a compensatory fermentation, leading to dysregulated cell growth. Very, very clear, very pointed. We just followed in the pathway of Warburg and looked deeply into what he was saying. And we've confirmed almost everything that Warburg was saying except we have shown that he was in error in trying to quantify the glucose pathway and the amount of energy coming out of the mitochondria from oxidative phosphorylation. He did not know about amino acid fermentation, another fuel that can also generate energy through substrate level phosphorylation, but it occurs in that organelle, the mitochondria. Now we know for sure how these cells are getting their energy, and we know how now to kill them, because once you know what they're eating, you know how to kill them. So and people are chasing genes. They're looking at all these mutations in the nucleus, trying to do precision medicine. All those genetic mutations are downstream effects of the abnormal energy metabolism. Okay, so that's why we're not making any major advances. We keep chasing things that are downstream and not central to the issue of what's causing the dysregulated cell growth. So constantly we're throwing good money after bad on all these fundraisers and all this stuff. And if you look at the scientific advisory boards, not only for the National Institutes of Health, but also for these various cancer, Susan Komen and all these others, they all have guys that think cancer is a genetic disease sitting on their scientific advisory board. Therefore, you know you're throwing money, good money after bad, because those guys don't understand the biology of the disorder they're treating.